This is a biology practical class for BIO111 General Biology Practical. What I'm going to be doing now is to give you a general introduction. This introduction is good for you before you enter the lab. There are things you should know as a biologist. There are things you should know as you enter into the biology lab. There are do's, there are don'ts that we are expected to introduce you to. And so for this class, a laboratory practical manual guide, a manual is a guide, is available. You will have to pay and pick a copy for yourself. Each student is expected to own a copy of the manual. And this is because all the practical work and report that we are going to be doing will be reported directly, will be done directly inside the manual. And so two students cannot share a manual. Manual per student, reporting per student, submission per student. Students are expected to have a practical drawing book in addition to the manual, because there may be not enough drawing spaces, and biology is all about a lot of it will have to do with drawing and with identification. And both the manual and the practical drawing books will once in a while be submitted for assessment. The basic content of this manual, basic content that we are going to see in the manual as we get it, is number one, you'll find on the first 1 or 2 pages some useful hints for using the practical manual. The manual itself contains a guide on how to use it. Every instrument that you buy in the market, whether your handset or your phone, your wristwatch, they contain certain information, a small leaflet that guides you on how to use it. So this manual has a guide on how to use itself. Some safety measures for using the laboratory are also included. The do's and the don'ts, what you will do or are expected to do in the laboratory, and what you should avoid. They are in the manual. Read them carefully. Where you don't understand, call the attention of the technologist, and it will be explained to you. We have general laboratory safety precautions. We should not be careless in the laboratory, and so we have made some safety precautions that you will observe. We have made them available. Don't eat in the lab. Wash your face quickly if something spills on you, and so on and so forth. Special precautions for working with heat or fire. They are also, we have set them they are there. We have provided those precautions. Those for chemicals, those for electrical equipment, and those for glasswares and other laboratory equipment, and then laboratory specimens, live or preserved. There is also a content for first aid in the manual. They are all provided inside that small manual. The manual is a small book that guides you page by page on what you will be expected to do. All the practicals will be expected to carry on in the lab. During the practical sessions, there are certain expectations. We have divided this practical into different sessions. There is section A of the practical is talking about laboratory apparatus, instruments, glassware, and solutions. These are together called laboratory apparatus. Laboratory apparatus, instruments, glassware, and solutions. One of the key instruments that we use in a biology lab is the microscope, and you will be introduced to carefully, detailing into this important. There are other instruments, but we will take time. We have taken our time inside this manual to explain the uses, the types of microscopes, the parts of a microscope. You'll be expected to know every part, how to use a microscope, and when the parts are not functioning, we'll be expected to know how to change them, because this is our major instrument that we use in biology, the microscope. It is the most frequent used instrument. We'll also be looking at how to prepare common laboratory solutions two types of solutions. There is one we call the stock solution. Stock solution. Stock means that a solution that we can prepare and keep it for a long time. That is why it's stock. And from this one, we can now be preparing the second one that is called a working solution. Working solution. So we can get a working solution prepared out of a stock solution. And the working solution, we use it over a small period of time, and even if it remains, we discard it and go back to the stock solution to make another working solution. We'll be looking at all that. Section B of this manual, we'll be looking at the cell and the cellular processes. Cell and the cellular processes. Under the cell, we'll look at the plant cell. We'll also look at the animal cell. Very important. Remember, we said biology is the study of plants and animals, though we also study the environment. There are some practical sessions that we'll be looking at under ecology about the environment. But for this, section B, we have given attention to the cells of plant and animal. The cell is a structural and functionally fundamental part of the organisms. And so we are going to be looking at the structure and functions of cell and the cell organelles. Structure, function of cells and their organelles. They are all contained in the manual. It will be important when we are asked, when we are required to draw a cell, we should be careful. There are rules guiding drawings. Sharp lines, clear objects, no shading, all the rules are there. And so, when we are asked to look at functions, carefully, every organelle has a function. So we'll be looking at all that under this structure and function of the cell. But cellular processes, there are 2 or 3 of them. The first one is diffusion. We also have osmosis, and also have, sometimes we have what we call plasmolysis. Diffusion, osmosis, and plasmolysis. But we'll pay attention. This book has given attention, the guide. We have given attention to diffusion and osmosis. And then, like I said, we are going to look at the section on ecology where we have interaction between the organism and the environment. Under this, we are going to be looking at several energy levels and feeding relationship. We'll be creating food chains. We'll be required to create food chains and create food webs. And we'll also be required to construct pyramids of numbers and pyramids of biomass. We'll be required to do mosquito studies, and then the different ways of studying the habitat. And then we'll be looking at how to do some control of mosquito vectors. So these are some key practicals that have been there, that are there in section C. In section D, we're going to introduce you to microbiology, some aspects of microbiology, studies of small, tiny living organisms that cannot be seen with the naked eyes. And in this case, we'll be looking at the effects of boiling on microbes. We'll also be looking at the effects of filtration on microbes. And we'll look at the ubiquitous nature of microbes, how they are found everywhere, in the air, in water, and in the soil. So we'll be doing some soil, taking samples from the soil, taking samples from water, and taking samples from the air to find out the presence of microorganisms. And section E is going to be talking about genetics. We'll be looking at certain chromosomes. I'll be looking at variations in populations in genetics. Chromosomes in plants, chromosomes, we'll be using the onion, for example. And we'll also be looking at, we have also created space for some chromosomal arrangements in animals and variations in populations. So that will be that for these lectures, for these practicals. The practical guide has these sections, and these sections will be properly guided. So you are now free to go to the lab and take your practicals under the guide of technologists. We will also make ourselves available, and if there are questions, we will answer them from the lab. I wish you a very good lab session throughout this semester. Thank you.